Hello everyone, welcome to my channel Online Biology Tutorials. In this video, we will study a very important topic from chapter number 6, Molecular Basis of Inheritance. And the name of the topic is DNA Fingerprinting. DNA fingerprinting is a very important tool for the forensic experts if they want to solve any mystery. For example, if murder happens somewhere or in some rape cases as well, if they want to find out who is the actual culprit, then this is the one technique which is going to help them out. So let's understand the basics first, and then we will study how it actually helps in finding the suspect. So we have already discussed the Human Genome Project, and in that we have already discussed that 99.9% we are similar to each other. Means I resemble to you and you resemble to me in 99.9%. We are this similar to each other only in 0.1%, and this repetition in the genetic material makes us distinct, makes us different from each other. Okay, so if we take DNA and if we do the density gradient of it, then we will get two peaks. Okay, we will get two peaks. Of course, the bigger peak will be the bulk DNA, and bulk DNA will be the one by which we resemble to each other. Means 99.9% we will get that DNA which is in huge amount and similar in every one of us. Whereas 0.1% will be that DNA which is in the form of repetition. Okay, so this DNA which is in the form of repetition, this DNA is known as satellite DNA. You must be remembering that in class 11th we discussed about the SAT chromosomes, right? So whenever you have to draw chromosomes, they are drawn like this. The central circular structure, it is known as primary constriction, and this primary constriction is also known as centromere. Okay, this is also known as centromere. Some chromosomes, beside containing the primary constriction, they also contain secondary constriction as well. Okay, so this secondary constriction makes this chromosome as the SAT chromosomes. Okay, so SAT means satellite chromosomes. So the chromosomes that have primary and secondary constriction both, they have SAT chromosomes, and in them generally SAT DNA is found. Now we will see further categorization, like in how many parts satellite DNA has been divided. One thing is sure, that satellite DNA will always be the repetitive DNA. So satellite DNA is basically divided into microsatellite and mini satellite. Now why is it divided into micro and mini satellite? Because the amount of purine and pyrimidine varies in them. Okay, the length of repeats is also varying in them, right? So these are few criteria through which we can segregate the repetitive DNA. Now if I basically talk about microsatellites, then this type of DNA contains 2 to 4 base pairs in repetition, whereas 20 to 25 base pairs are there in repetition in the case of mini satellites. Now if I say here in the case of microsatellite that the base pairs are 2 to 4 in number and repetition, for example, if I am writing CG, 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 here you can observe that CG is containing 2 nucleotides. So 2 nucleotides are repeated. These nucleotides are repeated, they are one after the other, and such type of repeats are known as tandem repeats. So in the case of tandem repeats, in the case of microsatellites, I may have CG, CG, CG and so on, or you may have CGA, and CGA makes 3 nucleotides. So you may have CGA, CGA, CGA. Your friend may have ATA, ATA, ATA. Okay. So this way repeats are different in my body to your body and to someone else. That's why we are different to each other. But the number of base pairs will range from 2 to 4 if we are talking about microsatellite DNA. Right? Now here, in the case of mini satellites, the tandem repeats will be 20 to 25 base pair long. Let's suppose here I draw a sequence. Okay, it is ATA, 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 CGG, GGC, CG. So here you can see till now I have made 17 nucleotides. So this is our 20 base pair long sequence, right? This is our 20 base pair long sequence. Now as soon as it will get finished, after that the same sequence will be there. Okay, the same sequence which I have written with green, this black sequence will be written in the same manner. Okay, so 20 bases will be here and so on, it will be followed. So here the tandem repeats length for about 20 base pairs. Okay, so this is happening in the case of mini satellite. You can see that the length of repeat, the number of tandem repeats, they are varying in the case of microsatellites and mini satellites, right? So overall we can conclude that in the case of microsatellites and in the case of mini satellites, there are variable number of tandem repeats. Okay, variable number of tandem repeats, and these VNTRs, which is the abbreviation of variable number of tandem repeats, these are very important because they are the basic tool for studying the DNA fingerprinting. Now we will study the steps which are involved for studying the DNA fingerprinting. So our next heading is going to be the steps for DNA fingerprinting. Okay, now we will write point by point. The first step is we need to isolate the DNA. Okay, so if there is any crime site, there must be some amount of DNA associated in the form of hair follicle. It could be some blood sample, it could be saliva, it could be sperm also. Okay, so all these samples, these are the samples from which DNA can be extracted. Remember, whenever we are isolating the DNA, we just need to check the amount. Is the amount appropriate for the following procedures? If the people involved in the DNA fingerprinting, they found that the amount is less, then they adopt a technique which is known as polymerase chain reaction. This polymerase chain reaction is abbreviated as PCR, and it is a specific machine into which all the requirements are fed and then DNA is taken out. So polymerase chain reaction actually increases the amount of DNA as per your requirement. You just have to feed in the requirements, how much of DNA do you require, then you give all the requirements to this machine and it will process it. Okay, so if the amount of DNA to process the experiment, to process the DNA fingerprinting is less, we need to go with the second step. Otherwise, if we have appropriate amount of DNA, we are not required to do so. Next step is going to be the treatment of DNA with restriction endonucleases. Okay, so DNA is treated with restriction endonucleases. These restriction endonucleases are the enzymes that will break the DNA in between, and it will produce varying fragments of DNA. Why am I saying varying? Because each fragment will differ in its length. Okay, then we will separate the DNA. DNA is separated by electrophoresis. This is a technique which will separate the DNA basing upon its length. Then the DNA fragments are separated, or they are transferred to nitrocellulose paper. Then it is fused with VNTR, means artificially variable number of tandem repeats will be designed and they will contain radioactive bases. So artificially designed VNTRs, they are known as VNTR probes, and in the end radiography will be done. So auto radiogram will be prepared, and this will give an impression like what is the DNA fingerprint of this individual. So in the end you will get a result which will show the bands like this. Okay, so in the end somewhat this kind of result will be seen, where a X-ray film will show that this is the DNA fingerprinting pattern. Okay, DNA fingerprinting is done and it will give a definite pattern. That means if I undergo DNA fingerprinting technique for my DNA, it will have a specific DNA fingerprinting pattern, and so as if you will go for. That's why every individual has a specific DNA fingerprinting pattern. Okay, now let's see how we can really find out if someone is involved in some mystery. Okay, so what we'll do, first we'll understand. Let's take the case of two individuals. Suppose one is Alexa and other is Jack. Okay, and variable number of tandem repeats, they are present on each chromosome. Generally SAT chromosomes are present everywhere in our human genome. So we are just taking about 2-3 chromosomes. We'll just discuss about 2-3 chromosomes. Okay, so we know that chromosomes, they exist in pairs. They are the homologous chromosomes, right? So we will discuss. Now whenever I am making a block, it means a tandem repeat. So here in the case of chromosome number 1, I have drawn homologous chromosomes, and you can see that the chromosome number 1, it is containing 2 tandem repeats. Okay, so these are my 2 tandem repeats, and here 1 means the chromosome number. Okay, now let's proceed and draw for chromosome number 2. Now if I am making it again, the blocks here means the tandem repeats. Okay, so here I have made 3 tandem repeats are there. Now if I take the case of Jack, suppose here we talk about chromosome number 1, and suppose Jack in the case of chromosome number 1 has 6 tandem repeats. Okay, similarly you will be making 6 blocks here, and suppose this Jack has 6 tandem repeats on chromosome number 1, whereas on chromosome number 2, let me take another color, and suppose on chromosome number 2 he just has 5 tandem repeats. Okay, so this is the pictorial representation of tandem repeats. Now, if I go for DNA fingerprinting in the case of Alexa, right, a specific pattern will be there, and the DNA, these tandem repeats, the smaller one will be there at the base, right? Then their number will consecutively increase. Similarly, for Jack, we will be doing DNA fingerprinting, and the numbers, of course, the smaller one will be near the base, and this way we need to move on. Okay, so here let's see first in the case of Alexa. She has 2 tandem repeats on chromosome number 1. So somewhere a band will be seen here in the case of 2 tandem repeats, right? Now let's move on to chromosome number 2, which has 3 tandem repeats. That means a band will be received here. So this is just the banding pattern or the DNA fingerprinting pattern of Alexa for 2 chromosomes. Similarly, she might be having other chromosomes with tandem repeats. So accordingly we can go on for the rest of the chromosomes. Now here for Jack, he has 6 tandem repeats on chromosome number 1. So it would be somewhere here, and the number of tandem repeats are more, thus the length of the banding pattern will be a little bit longer. Now if we see for chromosome number 2, it is 5, so it will be located here. So you can see that here we have done for Alexa and here we have done for Jack. Is DNA fingerprinting difference present? Yes, it is. DNA fingerprinting pattern of Alexa was different and Jack is 100% different. That is why it is useful in the forensic science. Now suppose, let's take one more case because you have understood now. Let's suppose we have got a DNA sample in the form of hair from the site of injury or site of accident, right? Then what will we do? We will separate the DNA. We will increase its amount because the amount of DNA which we'll be getting from hair would be very, very less. So we need to enhance its amount. We will be doing PCR. Okay. After doing PCR, we will be following all the steps like DNA will be done, electrophoresis, right? And then what we will do? We will get a specific banding pattern. Okay. So suppose we have got such type of banding pattern in the end from the DNA that we have got from the accidental site. Now there must be various suspects which will be involved in this accidental case or the murder mystery. Suppose we have 4 people involved and we say them as suspect 1, suspect 2, suspect 3 and suspect 4. Okay. Now we will be doing DNA fingerprinting of all these individuals, suspect 1 to suspect 4. Their DNA fingerprinting pattern would be specific, right? So we will match it with this one because this was the DNA fingerprinting pattern of the DNA that was achieved, that was received from the DNA at the accidental site. So of course this hair follicle would be of that individual who is involved in this murder mystery. Okay. So if suppose suspect 2 has the same DNA fingerprinting pattern to that found in the hair. Okay. So this will confirm that this person is involved. He or she has done this murder. Okay. Now if you must be confused that hair follicle or hair which has been received at the murder mystery site, it could be of the victim. So we can do one more thing. We can do DNA fingerprinting of the victim as well. If the DNA fingerprinting of the victim matches with the hair, that means that hair will be of the victim that was murdered. Otherwise, if the DNA fingerprinting of that hair does not matches with the victim, of course it will be of the suspect. Okay, so this is how it is useful to the forensic people. They just match the DNA fingerprinting patterns and then they come up with result. Hope this was helpful to you. Stay tuned for more videos. Thank you.